And this is chapter 3. This is part A for cells, the living units. Again, look at the PowerPoint before the lecture because I'm going to comment on the slides, not read them to you, but the content obviously is important as well. So here we go. Every body is composed of cells. And cell theory seems very logical at this stage of the game, but historically, people did have a time when they felt that cells actually came from dead material, something that had died. And now we believe in biogenesis, that cells can only come from other living cells. And they will then, in the human body, be able to collectively form tissues and organ systems of the organism. And this is just showing the various types of cells that you can find in the body, from blood cells to fibroblasts to fat cells to nerve cells to sperm, and all of them are composed of cells. We're going to talk at length about a generalized animal cell in the human body. And all human cells have these three parts. They have a membrane. And the outer plasma membrane is going to define the inside and the outside of the cell so that the cell has the ability to be different than the outside environment, but still communicate with that outside environment, but in a selective way. Inside is composed of a cytoplasm, which is kind of like a gel material in which organelles can be suspended and certain chemicals and atoms and molecules can be dissolved in. And then the control center, the nucleus, which houses the DNA, the chromosomes, the genes, which are the blueprints for being able to make everything in the cell. And this is a typical generalized animal cell, cell in your body. So kind of going from the outside to the inside, here is your plasma membrane, which we'll talk about at length in a minute. Here are mitochondria that make ATP. Here are rough endoplasmic reticulum, where protein synthesis occurs. You can then concentrate proteins, and that's going to be in the Golgi apparatus, so the Golgi body. Here are some vacuoles filled with proteins for import or for export. Then you have things like the lysosome, which is right here, a bag of degradative enzymes. I'm not going to go through each and every one of them, but of course here's the nucleus with the nuclear membrane and the pores and the nucleolus and the chromatin. The plasma membrane is kind of the crowning glory of the cell. It's composed of a phospholipid bilayer. Why lipid? Lipid repels water. And your body's 70% water, your cells are 70% water. If you want to have a selective membrane that will preserve the integrity of the inside of a cell versus its environment outside of a cell, you're going to need a molecule, or an organic molecule, like a fat, like a lipid, that's going to repel water. And that means that the membrane is actually composed of 2 layers of fat. And you have phosphate associated with these fats or lipids because the phosphate can interact with the water, so you have them facing out and facing in, so at least the water can touch the membrane, but the rest of it, then the fat is going to repel it. So very often we say that the phospholipid bilayer, the plasma membrane, is a fluid mosaic, and the fluidity is the fat. Believe it or not, these fat molecules are able to move like the ocean. And then the mosaic part of the phospholipid bilayer of the plasma membrane are proteins that are inserted into it that would be selective for transporting molecules in and out. So they're like ships on the ocean. So that's kind of the fluid mosaic part. And interstitial fluid is the fluid outside, and obviously cytoplasm is the fluid inside. So this is a depiction of, this is the phospholipid, these are the two layers of fat, and here are the proteins that are inserted, like ships in an ocean, that give selectivity to the cell as far as what goes in and out. So this will talk about a little bit about the types of lipids. The most prevalent lipid is the phospholipid, but believe it or not, you do have about 20% cholesterol. They come kind of like in plaques along the phospholipid bilayer, and cholesterol is actually the good guy in this case because it is able to make sure that if it's very hot out, you're not going to have the phospholipid bilayer melt on you, and if it's very cold, it won't get very rigid. So the cholesterol is there to make sure that the fluidity of the membrane is not compromised. There are these integral proteins. When you go back to that other picture, you can see some of the proteins are actually quite large. And these are transmembrane proteins, and they can function as transport enzymes or receptors. So they can have a number of different functions that would enable some regulation of what goes in and out of a cell, outside to inside, inside to outside. Peripheral proteins are loosely attached either to the outer membrane or the inner membrane, and they can function as enzymes, motor proteins, to change shape during cell division. Also, they can function as muscle contraction proteins. They can also be cell-to-cell proteins that will enable neighboring cells to attach or communicate with each other. And also, I'm not sure whether these animations are actually live or not, but if they're not, then looking at the diagrams or illustrations should be fine. Okay. So this would like be a peripheral protein, that would be an integral protein. And there are 6 functions of membrane proteins. The first is transport, and you will see a diagram in a second, but transport means that it's almost like a tube where but the specificity to the tube is not going to allow everything to go in and out, but very specific molecules would be able to easily go in and out of the cell. The second one is called a receptor for signal transduction. I'm not going to emphasize that right now because, I mean, I want you to know it, but we'll get into more detail in AMP2. But signal transduction, as the name implies, is allowing certain molecules to carry a signal for change. And the transduction means that the signal would bind to a peripheral protein, but cause a series of steps that would translate into change within the cell. So that's what's meant by receptors for signal transduction. They're signals that are trying to go across the membrane and change the inside function of a cell. The third function of membrane proteins, you can have proteins that would actually be in the membrane and attach to the cytoskeleton, and that would kind of preserve the integrity of the shape of the cell. And sometimes you have extracellular matrix. These are proteins made by fibroblasts, usually connective tissue cells, like collagen. And the collagen is made and it's transported outside between cells so that these connective tissue proteins can attach to cells and give them extra cushioning. There's lots of different things that extracellular matrix can do to enhance the function of cells. So here's an example of transport. As you can see, the little red balls are going in and out of the cell. These are the receptors for signal transduction. So the signal binds, but the signal is not allowed inside the cell, so the cell has to start across the membrane, transmembrane, transduction, a series of cascade that would go on inside the cell, which the signal would start. And here we have a protein that is going to be in the membrane, be in the plasma membrane, but also attached to cytoskeletal proteins inside the cell or collagen strands outside the cell. Okay, so now we get to function 4, 5, and 6. Enzymatic activity. There are enzymes usually on the interface of a cell that will catalyze enzymatic reactions. And there are also proteins that are going to be on the outside of the cell that will allow cells to bind one to another. And you and I are unique creatures, and we have molecules, again, on the outside of the cell that are proteins that are self, S-E-L-F. They are seen by your immune system as you, and that self-recognition proteins would allow the body to recognize foreign cells like pathogens, and they would also foreign organs, and then the immune system would be able to distinguish between foreign cells, foreign organs, and self. So this is a picture of enzyme activity where you have a little green ball become a red ball, the red ball become a blue square, and those are trying to show the enzymatic activity of the protein embedded in the membrane. Intercellular joining. So we'll be getting into that a little bit more specifically in the next few slides. But CAMs are an abbreviation for cell adhesion molecules, kind of like Velcro on the outside, so you can have cells stick to each other. And cell-cell recognition. So the cells are able, these little green balls are the proteins that the cell would recognize as self, and you want to have that not only to recognize pathogens, but you want certain tissues and certain organs to recognize each other, and cell recognition proteins can help you do that as well. Now, I'm going to get into the cell-cell between cells, the cell junctions. They are 3 types: tight junctions, desmosomes, and gap junctions. So these are going to be proteins on the outer membrane or outer surface of the cell membrane or plasma membrane that would allow cells to bind to each other. Let me explain each one as they come up. So the first one is a tight junction. As the name implies, it's going to be proteins that bind very tightly to each other cell to cell. And here we have an example of the cells lining the gastrointestinal tract. If you did not have these tight junctions made up of proteins that would allow the cells in the gastrointestinal tract to have an impermeable barrier and prevent fluids from moving, then every time you drank a glass of water, it would leak out into the rest of the body. Every time you ate a sandwich, parts of the sandwich would leak out into the body. It doesn't happen. The water travels through the gastrointestinal tract from the mouth to either urinary system, into the urine, or into the feces. It doesn't leak. And so tight junctions are incredible. That means the cells of the esophagus don't allow fluids to leak out, stomach don't allow fluids to leak out, small and large intestine don't allow fluids to leak out. And your tuna fish sandwich doesn't leak out either. And that's all due to tight junctions. And here we go, tight junctions. And that's their depiction of that. All right, desmosomes. I like this. Rivets are spot wells that anchor cells together at plaques, which are the thickening area where you're going to find these desmosomes. And it says it can be found in cardiac muscle cells. So I want to explain. This has not to do about leaking anything. This has to do about giving adjacent cells a lot of strength. Cardiac muscle cells. The heart is a pump. And it's going to have periods of intense contraction, so you're going to be able to force out massive amounts of blood to the rest of the body. And the cells, if you have these powerful contractions without desmosomes, without these rivets, you would have the cells eventually just tear themselves apart. The contractions are just so massive that you have to weld them together in order for them to have a forceful contraction but not injure the cell that is contracting so forcefully. So you want to have all the cells in the atria contract at the same time, and then all the cells in the ventricle contract at the same time, so that you can have a unidirectional flow of blood and have it be very robust. But you have to have these to help the cells that are in these forceful contractions that demand a unified contraction of the upper and then the lower chambers, not to be eventually destroyed by the force of contractions. And that is called the desmosomes. And here is that welding that goes on. It's not so much a problem of leaking a glass of water or leaking a tuna fish sandwich. It's more a matter of making sure that the cells remain intact even though they are undergoing forceful contractions. And then gap junctions. It's interesting. Gap junctions are bridges, and they are bridges between, and they're going to use cardiac cells. Again, they're going to use heart, because the heart can be auto-stimulatory. You have pacemaker cells. We'll talk more about them when we get to cardiac and AMP2. But pretty much everybody realizes that the pacemakers are the part of the heart that will spontaneously discharge an electric signal that then will be eventually translated into the heart contracting, the heart pumping. And the gap junctions are these bridges, and they are separate from the desmosomes. And the gap junction, the bridges, are so this wave of excitement that's started by the pacemaker can travel at millisecond speed. And that is exactly, when you think about the fact that the heart might pump 90 beats per minute, and those are going to be incredibly fast contractions in order to meet the oxygen and nutrient demands of every cell in the body. And to have that go more slowly would end up in the death of the organism. So these bridges, these gap junctions, allow these millisecond electrical impulses to travel at lightning speeds in order to enable the heart to beat that many times per minute. So here are these little bridges which would allow these gap junctions, which would allow the electric impulse. So I want you to think about the cardiac cells. They would have the gap junctions. They would also have the desmosomes. So the force of contraction wouldn't tear them apart, and the electrical stimulation by the pacemaker would travel at lightning speed. Okay, so now that we're out of junctions between cells, we are now going to talk about how do you transport molecules selectively in and out of cells. So there are passive processes, such as diffusion and osmosis, and there are active processes like active transport or movement of host cell membranes. And that's what we're going to talk about. This particular part A is going to go into the passive processes, and then when we get to part B, we'll talk more about the active processes. So the passive process is called passive because you don't need any input of energy in the form of ATP. And molecules are going to just travel high concentration to low concentration. That's what they talk about when they say a substance moves down a concentration gradient. It sounds very confusing and very, I don't know, scientific. But that sentence can be also very simple. It's just that you're going to move things from high to low concentration. That is down a concentration gradient. Active processes, which we'll talk about next, is going to be in need of an input of energy, an input of ATP energy in order to have it work. So we're going to do passive, which is like movement for free first. So we have diffusion or filtration. So simple diffusion means that you're going to have certain very small molecules go from high to low concentration, inside to the outside, outside to the inside of cells. Sometimes they will need a carrier or a receptor in order to facilitate that, and that's going to be facilitated diffusion. And another is osmosis, where you can't move the molecule, but you can move water. Filtration happens in the lung, where you just have the movement of respiratory gases like oxygen and CO2 go in and out. And that happens at the level of the capillary. Okay, here we go. So here we have diffusion, where you have a molecule X. It's very high on the outside, not so much on the inside, and X is small enough to be able to just move very quickly from high to low. Facilitated diffusion is when you need a receptor to move something a little bit larger. Again, high to low. Osmosis is going to be about moving water from high to low because the molecule itself is not able to be moved. So let me show you a picture of this. Here we have something that you might find in the chemistry lab, where you have a tube and it's filled with little red balls in water. And there is a membrane that is not going to let the little red balls move. But you do want to have equilibrium. You want to have the same amount of fluid and red balls in both compartments. So if you can't move the red balls, you can move the water. The water is going to move from high water concentration to low water concentration. And as you can see right here, you have more water here, less red balls, and you have so many red balls here and actually less water molecules. The membrane will allow water to move from high to low. So at the end, when it reaches equilibrium, look at the level of water. It has greatly increased in this area and greatly decreased in this area. What this means is that you have less water, and the amount of the little red balls is going to be the same with in this compartment as that compartment. That's with my little cursor here. This compartment, that compartment, because there's more water to dilute out the high red balls in this section. So the amount, the percent of red balls to water is the same in both compartments at the end of osmosis. So why is this important? This is important because of, let me get to this right here, red blood cells. Red blood cells are in plasma and they love equilibrium, especially the equilibrium of salt. The amount of salt in the plasma should equal the amount of salt in the red blood cell. If that happens, you say it's isotonic, the same amount of salt in and out. And that's a very happy red blood cell isotonic solution. Here's your happy little red blood cell, and the amount of salt in and out are the same. But very often what we eat, let's say I just ate a bag of really salty potato chips, and the amount of salt is really high. So I eat the potato chips, and the salt is going to eventually move from the digestive tract into the blood. So for a short amount of time, there's way too much salt in that plasma, and it is said to be too much salt is hypertonic. Too much of something, hyper. So there's too much salt in the plasma and not enough in the red blood cell. And the blood cell wants equilibrium. And it doesn't allow the salt to move, it allows the water to move, however. So in order to get the same amount of salt in and out, water leaves the red blood cell and it starts to shrink. You have kidneys, and the kidneys will eventually come to your rescue and make everything isotonic again, but there will be a short period of time where it is hypertonic and the red blood cells do shrink, and the amount of oxygen being transported is going to be compromised. In contrast, you could have a situation, this has happened with athletes, where they end up just replacing the water that they lose in sweat and not the salt. That has been remedied by all kinds of sport drinks. But prior to sports drinks, athletes had to worry a lot about not only being hydrated, but replacing the salt that they're sweating out as well. Why? Because if they just replace the water and not the salt, they will become hypotonic. The plasma becomes less salty. And again, the poor little red blood cell wants equilibrium. And the way that it will get equilibrium is to move water into the red blood cell to make the salt inside the cell less concentrated. And again, a red blood cell that has now swelling and getting bigger is not going to be able to carry enough oxygen, and that would very much compromise an athlete. So what you want to have is isotonic, but if the plasma becomes too salty, hypertonic, cells will shrink. If it becomes hypotonic, not enough salt taken in, then the red blood cells will swell. And in both cases, hypo, this red blood cell is not functioning at its max, and that's going to compromise the person. Again, you will have things happen in the kidney that will try to preserve as much salt as possible, but like in all cases, the athlete, if they become hypotonic, would have to consume more salt. And there are other regulatory mechanisms that will kick in, especially the thirst response and cravings for salt and things of that nature. But could an athlete have a finite period of time when it's too low and this red blood cells swell? Yes. Could you have a very salty dinner and have a transient period where it's a little bit too salty in the plasma? Yes. So osmosis to the rescue as far as getting it equal, but you don't want to compromise the function of the red blood cell. And that's really one of the big reasons why osmosis is important in the body. So this is just a kind of a summary slide. You can have simple diffusion, molecules small enough to go high to low concentration. You can have facilitated diffusion where the molecule is just a little bit too big and needs to be transported by a receptor in and out. But again, it's for free. And osmosis where you have a situation, especially in the plasma, where the salt concentration can get too high or too low. And osmosis can reestablish equilibrium, but you then do have to worry at times about the proper functioning of, in this case, the red blood cell. Okay, so that is the end of Chapter 3, Cells, Part 1, and I will see you for Part 2.